Okay, guys, so Lac Operon. Lac Operon is a special operon present in the DNA of E. coli. Operon, as I told you earlier in the previous lecture, the link to which is given in the description, that operon is a functional unit of DNA that contains a cluster of genes which are sequentially arranged with the DNA regulatory sequence, the cis-acting elements of the DNA. If you don't know what cis-acting elements of the DNA are, so I would recommend please do check out the previous lecture before viewing this lecture. All right, so now Lac Operon, the name comes out from the disaccharide lactose. So Lac Operon is actually an operon that contains genes which code for enzymes that are involved in the metabolism of lactose. Basically, to be more specific, the catabolism of lactose. Okay, so it is a special operon of E. coli, a prokaryote that codes for enzymes. Basically, it contains three genes, namely LEC-Z, LEC-Y, LEC-A. These are the names of the genes. It contains three genes which code for enzymes that are involved in the catabolism of the disaccharide lactose. As these three genes are present in there. Okay, LEC-Z, it codes for an enzyme known as beta galactosidase. Okay, this actually breaks down the lactose into glucose and galactose. It actually catabolizes the lactose. Like Y, it goes for permease. This is an enzyme that facilitates the movement of lactose into the cell. So basically, it helps in the facilitated diffusion of lactose from the environment into the cell, into the bacterial cell. Like A, it goes for an enzyme known as thiogalactoside transacetylase. Its function is actually acetylation of the lactose group of the lactose sugar. So acetylation, basically addition of the acetyl group to a lactose. Well, its physiological function is unknown though. Okay, so but you guys should know what it goes for and what's its function. Okay, its function is acetylation, as its name suggests, thiogalactoside transacetylase. Right. So these three genes, they code for these three proteins, the three enzymes, and they are only synthesized. Okay, so this makes it very smart that these enzymes, the energy which is needed to synthesize all these enzymes, is only utilized, or the energy is only used in synthesizing these enzymes if lactose is available in the environment. Okay, so because lactose is the substrate on which these enzymes will work. So if lactose is not there, there is no point of producing these enzymes because it would just be a waste of energy. So these proteins, in order to conserve energy, in order to conserve the ATP production, they are only produced when the lactose, the substrate, is available in the environment. Okay, and now if this bacterial cell is given a choice between lactose and glucose to act as a respiratory substrate, it will choose glucose. Okay, because glucose is more rapidly metabolized and much higher yield of ATP is produced by metabolizing glucose rather than lactose. So it's basically the lactose is actually a slow sugar. It produces energy but pretty slowly, whereas glucose release is an instant source of energy. It produces energy very quickly. All right, so therefore bacteria, it uses glucose instead of lactose if glucose is available in the environment rather than lactose. All right, so it will not waste its energy in producing all these enzymes and then metabolize the low energy producing reagent, low energy producing substrate, but rather it will go for the much energy rich substrate, right? Glucose. And actually lesser energy is needed in metabolizing it because it's already in the metabolized form, in the metabolized state. It's already in the end product, glucose, whereas lactose has to be broken down into glucose and galactose, and then they have to be metabolized, right? So this is again an advantage of glucose to be used directly. So the bacterial cell is smart enough to save its energy by acting directly on the glucose molecules rather than acting on the lactose molecules. Now let's take a look at the Lac Operon structure. This is how it looks like. All right, so what is this? Well, this is actually pretty simple. Just these LEC-Z, LEC-Y, and LEC-A, these are the three genes which are present in this operon. These are the three genes that are sequentially arranged. And if these are the genes, what is the other component of an operon? It is the other component of an operon is the regulatory sequence of the DNA. This color over here, these three blocks, they represent the regulatory sequence of the DNA. Regulatory sequence of the DNA. Right. So now, in the regulatory sequence, we have The operator region, we have talked about the operator, what operator is and what's its role. It is the site where the repressor molecules come and bind, right? And it is arranged upstream of the three structural genes. Okay, these are the three structural genes. They code for the three enzymes. Like Z, as I told you, it codes for the enzyme beta galactosidase. Lac Y codes for permease, and Lac A codes for transacetylase. And then this operator is where the repressible molecule binds, the repressor molecule binds. All right, so it's the repressor binding site, and right upstream of the operator is your promoter region where the RNA polymerase binds. And then over here, there's an additional site as well, which is known as the cap region, the cap site. And we are going to talk about the cap in detail in a bit, right? So just hold on for cap. What is this cap over here? It looks like a cap on top of these mRNA molecule. So let's see what it is. Okay, so moving over here, this regulatory sequence of the DNA, which is upstream, there is a, okay, so I've told you what this is. The regulatory sequence of DNA is upstream of the three structural genes. Basically, they are talking about the operator region, the promoter region, and the cap region. Okay, that includes the promoter region, promoter site, where the RNA polymerase binds, the operator site, where the repressor molecule binds, and the additional site called the catabolite activator protein site. This is the cap site. Okay, cap is an acronym for catabolite activator protein site. Okay, or the cap site, catabolite activator protein. Okay, so at this point over here, catabolite activator protein, a complex of the CAMP, cyclic AMP, and this catabolite activator protein itself. This is a molecule which is present. Catabolite activator protein, when combines with cyclic AMP, it forms a complex called CRP, called CAMP regulatory protein, or simply this just the CRP. Okay, so now this complex, it goes and binds at the cap site, and if this complex is bound at the cap site and the operator region is empty, only in that condition will the RNA polymerase be permitted to initiate the transcription. So now let's go into a bit more detail. The regulatory sequence of DNA is upstream of the three structural genes that includes the promoter site. We've talked about it, where the RNA polymerase binds. Guys, please, you need to learn this function. Operator site, where the repressor molecule binds, an additional site called the cap region. Okay, at the operator and the cap sites, these are the two sites where the regulatory proteins bind. Okay, at the operator site, as I told you, the repressor molecule, and the cap site, this complex, CRP complex, and they are essentially important for the regulation of gene expression. Now, the genes are maximally expressed when the operator site is empty. Okay, no repressor must be present at the operator site, and the cap site is full. The cap site is occupied by this complex of CAMP and CAP. The complex of CAMP and CAP is known as CRP, CAMP regulatory protein. Now, the regulatory gene, okay, another regulatory gene which is not a part of the Lac Operon. I repeat, this is not a part of the Lac Operon. It has its own specific operon. This gene is known as this is a regulatory gene called the Lac-I. Now, this Lac-I, it codes for a repressor protein that binds to the O site with very high affinity. Right, so this is your regulatory protein, regulatory and molecule regulatory protein, which is produced by this Lac-I molecule, which has its own specific operon. This regulatory molecule, after being produced at that specific operon, acts as a trans-acting molecule, and it comes and binds at the operator, at the O site with very high affinity. Okay, so once this regulatory molecule, this repressor molecule, which is produced by the Lac-I, it binds at the O region, then you guys know that the RNA polymerase will not be able to initiate transcription. The Lac-I has its own operon and is not a part of the Lac operon. Right, so this is again your second regulatory molecule, regulatory protein, which is produced by the Lac-I gene. Right, Now, to sum up stuff, we've talked about the three structural genes: the operator region, the promoter region, and the cap site, the catabolite activator protein site. This is catabolite activator protein site, but at this level, at this site, the catabolite activator protein and the CAMP, they combine, they form the CRP complex, and this CRP complex, it comes and bind at the catabolite activator protein site. And so if we want if this transcription needs to be activated, this cap site must be full. It must be bound by the CRP molecule, the CRP complex, and the operator site must not be bound by the repressor molecule. The repressor molecule must not be present at the operator site. So now, if these two conditions are fulfilled, what will happen? RNA polymerase will come and attach to the promoter site. And once it attaches to the promoter site, then transcription will begin. Transcription of the structural genes will begin, giving a polycistronic mRNA molecule, and then that polycistronic mRNA molecule will be transcribed into the three genes which are needed, into the three proteins which are needed in the metabolism of lactose. Right. So this was what we did up till now. Now moving forward. Now there are specific conditions where, as I told you in the previous lectures, that Lac Operon is an inducible operon, so it can be easily regulated in presence or absence of certain molecules. Now let's look at them one by one. If lactose is not available, right, lactose is completely absent, then what happens? This Lac Operon is completely repressed. As I told you earlier in this lecture, that this is beneficial for the bacterium because it helps it save energy. So if lactose is not available, the Lac Operon is repressed. The inhibitor, which is coded by the LAC-I, LAC-I, as I told you, is another regulatory protein. Sorry, not a protein, regulatory gene, which is present at a different operon. It binds to the operator site. The inhibitor, which is coded by this gene, it comes and binds to this operator site. And once it binds, as I told you, that RNA polymerase will not be able to transcribe the structural genes. Then what happens? The repression protein binds. Okay, this inhibitor over here, a repressor protein, it comes and it binds to this operator site through a special supersecondary structure, through a special motif known as helix turn helix. Well, helix because there is an alpha helix, then there is a turn, and then there is another alpha helix. Okay, and this entire example, which is over here, the repression by this inhibitor, by this repressor molecule coded by the LAC-I, is known as negative regulation. And we've talked about negative regulation in the previous lectures as well, and I'll try giving the link to that as well in the description. Do check that out as well. All right, so negative regulation, because the presence of this repressor molecule, it prevents, or it actually represses the expression of the Lac Operon. So this is known as negative regulation of the Lac Operon. Now, I've given a structure over here of that motif, of that supersecondary structure, helix turn helix. Okay, motif is a supersecondary structure which contains more than two secondary structures that are arranged in a configuration that is stable. Okay, it contains one supersecondary structure, the alpha helix, then a turn over here, and then another alpha helix. Okay, this is the supersecondary motif from which is bounded to the operator, and then it is actually the repressor molecule comes and binds to the operator through this motif. Okay, now, so this was one case. When lactose is not available, it's completely repressed and it will not be expressed. The gene will not be expressed, and this will be beneficial for the E. coli because it will help it save energy. And when lactose becomes available, now the series of steps that occur. When lactose is available, it is converted into an isomer. Okay, this is important, guys. First, it is converted into an isomer known as allolactose that binds to the repressor, which is attached to the operator site already, okay, that repressor which was produced by the LEC is already attached to the operator that prevents the expression of the Lac Operon. But once the lactose becomes available, it is converted into allolactose that goes and binds to the repressor which is attached to the operator. The repressor and allolactose binding, it causes the shape of the repressor molecule to change. A conformational change occurs. Sorry for the spelling over here. A conformational change occurs, right? So basically, the conformation of the repressor molecule changes, and due to the change in conformation, the repressor molecule is unable to remain attached to the operator site, and hence it detaches it because they are specific to the shape. So after the operator molecule, it is free, it is empty. What happens now? This one condition of transcription initiation by the RNA polymerase is fulfilled, that the operator site has become empty. If it's empty, there is an enzyme called adenyl cyclase. Adenyl cyclase, okay, that enzyme is actually, if only lactose is present, okay, no glucose is present in the environment, this adenyl cyclase enzyme is active. In that case, it catalyzes the formation of the CAMP. The cyclic AMP is produced in presence of this enzyme. Now, once the cyclic AMP is formed, it combines with this cap molecule, the cap protein, and forms that complex which is needed, the CRP complex, the catabolite regulatory protein, the CAMP cap complex. Catabolite activator protein, now it is converted into catabolite regulatory protein by combining with the cyclic AMP. Sorry, not the catabolite regulator protein. It is converted into CAMP regulatory protein. Okay, the C stands for the CAMP. Now, once this complex is formed, it also binds to the cap region. As soon as these two conditions are fulfilled, RNA polymerase comes and it binds to the promoter site, and transcription is initiated. All right, so this is how transcription is initiated once lactose becomes available, and only in presence of lactose when no glucose is present. Now let's talk about a few specific cases. Okay, what happens if glucose is available and lactose is not? As I told you earlier, that to save energy, the Lac Operon is not activated, it is repressed so that the enzymes are not made. This cap protein, the catabolite activator protein, it needs the CAMP in order to bind to the cap site, okay, to form the complex and then bind to the cap site. But in presence of glucose, the adenyl cyclase enzyme is inactive, okay, it is not active, it is inactive. So if it's inactive, then the cyclic AMP will not be formed because this enzyme is needed for the formation of cyclic AMP. And if it is not formed, then CAP will remain unbound and will not be able to bind to the cap site. And if it will not be able to bind to the cap site, then this will stay repressed. Right? Now, it is inactive in presence of glucose. And this inactivation of the adenyl cyclase in presence of glucose is known as catabolite repression. Okay? Catabolite repression. Okay, so basically this is due to the adenyl cyclase is inactive in the presence of glucose, and the cap is not bound to the to the cyclic AMP. So it's due to the repression is due to this cap molecule in the unbound state. This type of repression is known as catabolite repression because the catabolite activator protein is not active. And the repressor molecule, it comes and it is bound to the operator site as well. So both the conditions are not fulfilled. Okay, for transcription to be initiated, the operator must be empty and this cap site must be full with the complex that is formed by the cap site. So this is what the condition, this is the condition in presence of glucose and in absence of lactose. Now let's reverse the conditions. What if glucose is not available and lactose is available? This is what we just spoke about. And this is a chart taken from your from your book, Lippincott Illustrated Reviews. So you can check that out, right? So what happens in that case? The CAP, the cap molecule, it binds with the CAMP, forming the CRP complex. The CRP complex binds to the cap site, and this is bound. This binding fulfills the first condition. Then this repressor molecule, which was present initially at the operator site, it binds to the allolactose, the isomer which is formed from the lactose molecule. And this allolactose and the repressor binding, it induces a conformational change, and it removes the repressor molecule. And once it is removed, then your RNA polymerase can bind to the promoter region and then start out and initiate the transcription of the three genes, and hence can produce the three enzymes which are needed for the metabolism of the of the lactose. Now the last condition, when both of them are present. Okay, what happens when both glucose and lactose are present? In this case, due to presence of, okay, if lactose is present, it will be converted to allolactose and it will bind to the repressor molecule, and the repressor molecule will be detached. Okay, this condition will occur. But now, what has happened? Glucose is also present. I told you earlier that if glucose or lactose are present, glucose will be given preference. So how is this preference achievable? How is it achieved? Well, I told you earlier that this glucose, it acts by inhibiting this enzyme adenylyl cyclase, and catabolize repression occurs because the catabolite activator protein is unbound. It is not able to bind to the cyclic AMP, which was to be produced by this adenylyl cyclase enzyme. If it is in the unbound state, it will not be able to form the CRP complex, and if it is not able to form the CRP complex, then the CRP complex will not be able to bind to the cap site. And if the CRP complex is not able to bind to the cap site, then this RNA polymerase will not be able to bind to the promoter and will not be able to efficiently synthesize this, will not be able to efficiently initiate the transcription process, will not be able to efficiently transcribe the polycistronic mRNA. Okay, so this is what happens in presence of both glucose and lactose. So this was pretty much it about Lac Operon. Okay, this is a paragraph taken from your book, Illustrated Reviews of Lippincott. You may read it out from your book. Okay, so I'll just go over it once. When both glucose and lactose are available, in this case, the Lac Operon is uninduced. Okay, I told you that it's uninduced. The reason is because the CRP is not formed, and then the CRP complex is not able to bind to the cap site. Okay, although one condition is fulfilled, but the other is not. And reason, because the adenylyl cyclase enzyme is inhibited. Okay, and transcription is negligible even if lactose is present at high concentration. Okay, reason, because the adenylyl cyclase enzyme is inhibited in presence of the glucose molecule. This process is known as catabolize repression, because the catabolite activator protein is not able to bind to the cyclic AMP. Now, due to catabolite repression, the CRP complex will not form, the CAMP-CAP complex will not form, and so cap site remains empty. If the cap site remains empty, the RNA polymerase is unable to effectively initiate the transcription process, even though the repressor is not bound to the O site. Okay, as I told you, lactose is present, so it will bind to the repressor molecule and it will induce a conformational change, and it will remove the repressor from the O site. But since this complex is not available, the CRP complex is not available, the three structural genes of the operon are only slightly expressed at only low basal levels. Okay, so that's it for the Lac Operon. Thank you so much, guys, and we'll be discussing the trip operon in the next lecture.